From the Australian Bureau of Statistics, the third leading cause of death was dementia and Alzheimer's disease. Along with that, the 14th leading cause of death was intentional self-harm, which we can see is a pretty big deal in Australia. So how can optogenetics solve this problem? So how does optogenetics work? So say we take a phospholipid bilayer, which is essentially our phospholipids, point inwards. These are what make up our membranes that determine outside the cell and within inside the cell. It's amphipathic, meaning that on the outside it's polar and on the inside is nonpolar. So the fatty saturated carbon chains remain on the inside and the polar ends remain on the outside so they can bind with water and whatnot and so on. This protein in the middle is called a G-coupled protein receptor. It's G-coupled because there's a G protein here. This is typically found in a lot of signaling as the normal system. Use yellow. A molecule will come in and bind, and this is some transport device will allow something through, or will begin a reaction, will ultimately send a signal off, and a cascade of reactions occur. So what we do in optogenetics is alter this GCPR in such a way that it's sensitive to light. And how we do that is, so we change the DNA structure, and we find the DNA from things like from other organisms that do have these things, and we can use that and implant it in, and ultimately change it. So that's another issue, but I'll explain this for now. So now with our new membrane, this is a new membrane. This is our what's known as a rhodopsin. And rhodopsin simply means rose light in Greek, meaning that these things are functional by rose light, but it's altered by other wavelengths. So you can pick specific wavelengths that make this receptor functional. So say we've altered this protein so that light can affect it, and instead of some molecule coming in and changing this, use yellow. Instead of this molecule coming in and changing this, and ultimately affecting it so something goes through and a reaction begins and whatnot, instead this is affected by light. I'll use green for light in this case, anti-intuitive. At a particular wavelength, some wavelength A, nanometers, whatever, this will induce the same effects as that molecule. So we can simulate reactions that require a specific molecule without the need of actually using that molecule. So we can just use light to induce the same effects, and we can get some pretty cool results from it. So this paper runs through the benefits of optogenetics in psychiatric disorders. And one of the examples they provide is through addiction, and how optogenetics can provide a useful tool in remedy of addiction. This is in relation to cocaine addiction. So, under the influence of cocaine, what happens is, here's the amygdala, hippocampus, medial prefrontal cortex, and ventral tegmental area, and your nucleus accumbens. These cells here are made up of medium spiny neurons, which are typically dopamine receptors, so D1 in this case, and is in control of the reward system. So that feeling of reward comes from this area. So projections of dopamine labeled by this green pathway onto the nucleus accumbens will give you that sense of reward. So under the influence of cocaine, at this location here, there is a blocking of the dopamine transporter. So dopamine builds up in this area. You get this strong sense of reward. Not only that, the hippocampus, linked by a glutaminergic pathway, provides context to the situation from memory. This will provide our influence, motivation, the medial prefrontal cortex. Whoops. It's a little bit messy. And the basolateral amygdala provides emotional valence. So if you have a cocaine addiction, what happens is that, say you've gone off cocaine and you've been addicted for years, the plasticity of these membranes change. By plasticity, it means that they morphologically change in such a way that they adapt to the environment they're situated in. And although addiction is still one of those things that's still being heavily researched and there aren't exact links, there is some evidence to say that the medial prefrontal cortex has a key role in addiction. So deep brain stimulation of the medial prefrontal cortex has provided a good tool to remedy addiction. Although it is an issue as it requires around 100 hertz. Whoops. And within these areas, because it's an intracranial stimulation, it's within these areas, 12 hertz is enough. Capital H. So this can be quite damaging, this 100 hertz, to everything surrounding this area. It's everything within. So Optogenetics offers us a new tool. And I'll explain what they do in a second. Yeah! This graph, say this is our medial prefrontal cortex. This is the NAC. Oh, not enough room. And there's a glutamatergic pathway linking to it. This is a ventral hippocampus here. Not completely necessary to have it in, but just for the sake of completion. Basolateral amygdala. These all provide projections onto the nucleus accumbens. And there's also the ventral tegmental area. Provides our change color. Looks cooler. Here. Dopamine pathway. So dopa. Mean. So at this area here, the glutamatergic pathway, this membrane here, it's altered, well, in this method, it's altered in such a way that it induces what's called long-term depression. So the membrane at this location, we'll just change this around, remove this. Whoa. So this membrane, it's been optogenetically modified so that this protein, there's actually quite a few of these proteins modified, but at this location, is functional by light. So by applying or adding in a luminopsin or some form of tiny LED into the brain, say this is light, activates it, and positive, negatives go through, signals cascade. So by adding in rhodopsin here, changing this protein to rhodopsin will allow us to change the signal on our potential graph. So on the graph that we saw previously with red, let's go crazy and use this color. I want a black. So on our graph that we saw earlier, whoops, this is time, this is our governed threshold. By changing the plasticity of this pathway, it is shown that this has been reduced. This threshold, if this is an abnormally high threshold, and we want to get this back down to normal, N for normal, by changing this glutamatergic pathway, and altering it so that signal from the medial prefrontal cortex, whoops, there we go. So the signal from the medial prefrontal cortex via glutamatergic. Ran out of room. Pathway. It's decrease the signal so that now normal daily activities will reach over that. It's acting really slow. And will actually receive the signals that are above this threshold, A to B, not these ones. So that's what's known as long-term depression, because this goes down. And this is a lot easier with optogenetic control, considering that deep brain stimulation, as soon as you stimulate this particular membrane, there's a lot of variance in what's happening. And say this is outside of what's happening. This is going in the brain. There's a lot of variance as soon as this goes in. And this signal, you don't have much specific control, site-specific control. And optogenetics allows for site-specific control, i.e. meaning that you've mutated these proteins in such a way that you know these are active by light. These proteins on the membrane that allow for particular ions to go through or out, say negative, you know you have full control on these tagged proteins here. Because you've inserted the RNA, you know where it's going, you know what's happening. So what we found is that when you manipulate the medial prefrontal cortex at this glutamatergic site here, this change in potential after long-term depression. So this isn't under cocaine. So when you're not taking cocaine, the plasticity changes at this site, and you need to bring that down. You need to bring that threshold down. So this isn't when cocaine is within the system. So from this paper, they found that when they dissected the nucleus accumbens, that long-term depression was evident. And I mean they had strong evidence to say that this threshold has been brought down. So what is the future of optogenetics? Well, there is still a long way to go, but the best part is designer drugs. So in the pharmaceutical industry, designer pharmaceuticals will essentially allow us to take a pill that provides us with the RNA / maybe DNA that will mutate and translate to the proteins, to the rhodopsin and the luminopsin. Whoops, not enough room. Just get rid of this. Luminopsin that we need to provide support on our mental illness. So things like Parkinson's disease, post-traumatic stress disease, work in a very similar way to addiction. So it's a matter of targeting those areas in the neural networks, the particular cells, and altering them such that they're susceptible to light and they have function by the light, and providing proteins or LEDs, light-emitting diodes, to that specific area that can control these rhodopsins and allow particular signaling, either depression or high signal transduction, either way. What's interesting is that there was another experiment done on mice, which essentially they put these mice under a, this is my bad picture of a mouse, sorry. They put these mice under a fear stimulus and tagged the areas in the hippocampus. Say if this is my hippocampus, tagged areas, hippocampus, where episodic memory ingraining is, so episodes such as this fear stimulus, and they changed these channeling proteins to rhodopsin. So undergoing this change, when the mice were at a normal state, they either, I'm not too sure, they didn't really specify at this point, they introduced a light onto these channeling proteins within the membrane, which activated this fear stimulus, and you could physically see it in the mice because they froze up, they became cold and stiff, which is another example of how you could control specific emotions like this and ultimately get rid of things like depression, because maybe you could introduce a happy environment and allow some happy stimulus, and that could save this happy stimulus. Not really sure what I was drawing there. This could provide some good remedy to depression, as you could target this and have it programmed in such a way that it knows when there's a fear stimulus based on the way the networks are corresponding around it. And that way it could activate these rhodopsins and provide a happy stimulus and stop the person from potentially inflicting self-harm. Anyway, that's it for the presentation, and thanks for watching. Just write thanks, just in case. And Finn to sign off. Cheers.